So we started with the nervous system. We talked about the idea that the nervous system is broken down into two main parts, your central nervous system and your peripheral nervous system. So what does your central nervous system consist of? What are the parts? Right, your brain and your spinal cord, that's your central nervous system. And then the peripheral nervous system then is everything else, essentially, at that point. And then within your peripheral nervous system, you can break that down into two parts, the afferent and the efferent part. And so the afferent are your sensory nerves. So that's the stuff that information gets sent back to the central nervous system from your extremities or wherever those sensory things are going on. Efferent are motor nerves, so that would be signals that are sent from your central nervous system down to your peripheral nervous system, which makes muscle actions and those sorts of things actually occur. And then within your motor nerves, the efferent system, then you have your somatic nerves and your autonomic nerves. What's the difference between those two? Right, your autonomic nervous system, they kind of control themselves. And so that's like your digestive system, respiration. Those are things you don't necessarily need to be in conscious control of, whereas your somatic nerves are the ones that you're in conscious control of, where you have voluntary muscle actions going on with that stuff. On your exam, I'll probably ask you to draw for me a neuron and then label different parts of it. So what are the dendrites? What are their function? Right, they're the tentacle-looking things that collect information from other neurons and things of that nature. So they bring information in to the neuron itself. And then the axon does the opposite, right? So the axon is what sends a signal out from the neuron itself, and there's just the one axon. The soma is, what's the soma again? That's the cell body, right? That's the main kind of part that the dendrites are sticking out of, if you want to think about it that way. The myelin sheath, then, is what covers the axon, and it's what helps make the signals be fast and coordinated as they're sent out from a neuron to wherever it's sending those messages to. Then the terms polarized, depolarized, and repolarized all refer to states of the axon. So what does the polarized state refer to? That's the negative charge, and that's what it's at rest. And so your neuron is at a polarized state at rest. It becomes depolarized when a signal is being sent down it, which means it goes from a negative charge to a positive charge. And then being repolarized means it goes back to a polarized state. So that's the reset system, essentially, with that. So where do the chemicals sodium and potassium come into play then?
So it flows into the axon to charge and decharge it?
Right. So sodium's main function is that it's allowed to flow into the axon to make it go from a negative charge to a positive charge or to become depolarized. And then it has to be pumped out. Sodium has to be pumped out in order to kind of reset it or to repolarize the system. And then potassium is also essentially allowed to leave those little areas of your axon in order to have it go back to a negative charge. So the synapse is what then?
It's the space between the axon and the next dendrite or whatever.
That's right. That's right. So at the very end of your axon, the gap between it and whatever it's communicating with, whether it's a muscle or another neuron, is called your synapse. And so that's that little gap that is kind of in place there so that the signal doesn't get confused. So the synaptic vesicles, then, those are the little, we call them kind of little bubbles that are basically located at the end of your axon. And the synaptic vesicles carry your acetylcholine, which is your neurotransmitter, and then they transfer the acetylcholine to the edge of the synapse, and they allow acetylcholine to drift across the synapse, then communicate with whatever's on the other side of the synapse to propagate that signal that is being sent down that neuron. So the terms presynaptic membrane would just be essentially anything before the synapse. The postsynaptic membrane is whatever's being picked up on the other side by the other neurons, dendrites, or the muscle, or whatever it is that we're looking at. What do the terms temporal and spatial summation refer to then? Right, and so we kind of said that according to the all or none law, basically says a neuron either sends a signal or it doesn't. There's no such thing as a strong signal or a weak signal, but in order to kind of make a signal be seemingly stronger, you can have temporal summation, which means a neuron just repeatedly sends that signal over and over and over again to make sure that the muscle contraction is sustained or whatever it is you're trying to do. So temporal summation refers to a signal being sent repeatedly by one axon. And then what's spatial summation then? Right, that means you have several axons or several neurons sending signals simultaneously, and so they're all kind of joining in to send that signal to make that that muscle action or whatever signal is being sent appear to be stronger, essentially. Right. So that's your basic nervous system kind of things before we get into the reflex actions. Questions on that? That stuff should be pretty good for you guys because it's not the first time you've heard a lot of those things, so that hopefully should be pretty easy for you. All right, so for your reflex actions, this is where we got into kind of the ways that your body can control signals that are very, very fast. So the two reflexes that we talked about were the stretch reflex, and then the other reflex was reciprocal inhibition. Those are the two reflex actions we talked about. So using the terms that are up there, what is the stretch reflex? Yeah, it involves your muscle spindles. In what way then? What are your spindles responsible for? What do they do? Right, they detect a change in length, so of a muscle fiber essentially. So the muscle spindles are located inside and alongside all the muscle fibers. That's where they're located. And so anytime there's a change in length, the muscle spindles are activated, and then they send a signal to your spinal cord. And they indicate that the muscle's lengthening or the muscle's shortening or whatever it happens to be. And so they're considered a proprioceptor because they're detecting motion, essentially, around your body. And that's kind of that broad category of these things that we're talking about here with muscle spindles and the Golgi tendon organs. So the spindles detect changes in length. What do the Golgi tendon organs detect?
Tension.
Right, levels of tension, right? So they're located in your tendons. That's the only difference between those two. Is one detects tension, one detects muscle length changes, and then one's located in your muscles and one's located in your tendons. So the muscle spindles are involved in the stretch reflex, and the Golgi tendon organs are involved in reciprocal inhibition. So what do those two reflex actions cause to occur? What happens with the stretch reflex? What's that?
Do they get stronger?
Do they get stronger? Sort of. No, that's the role of the reciprocal inhibition.
There's stretch contracts muscles to prevent injury.
That's right. So a muscle, all of a sudden, the muscle will lengthen. The muscle spindles go crazy. They send a signal to the spinal cord. The spinal cord sends a signal back to that muscle and causes it to contract because it's a protective mechanism to keep the muscle from stretching too far and then rupturing. So what's nice about the stretch reflex, as far as we're concerned in terms of performance, is that it can actually make you perform better because it makes that muscle contract, which helps you jump higher, helps you throw harder. Those kinds of things can actually increase your performance. Reciprocal inhibition does what Megan was saying, is that it's designed to help reduce the tension in that muscle because when the tension becomes too high, again, as a protective mechanism, it tries to reduce that tension. What two things happen in order to reduce the tension as part of reciprocal inhibition? This involves these two other terms that are up here, agonist and antagonist.
Relaxes the agonist,
Mm-hmm.
contracts the antagonist.
Right, so the agonist muscle's contracting, and when that's contracting, then it's making the tension increase in that tendon. And so it detects that level of tension, sends that signal back to the spinal cord. Reciprocal inhibition then causes one thing to happen is going to be the relaxation of that agonist muscle. So it starts to relax, which reduces the tension on that tendon. And simultaneously, then the antagonist muscle starts to contract because that will also reduce the tension around that tendon. So if you think about this in terms of your muscles of your quadriceps and your hamstrings, they're antagonists to one another. And if you think about the tension around your patellar tendon, if that tension gets really, really high in your patellar tendon, the way to reduce it is to relax your quads and contract your hamstrings, and that will bring that tension down. And so we kind of saw that a little bit when we did some of the different, when you guys jumped using off the box and you jumped without using the box. And we saw, I think it was Jonathan maybe, when he jumped off the box and landed, it was just like, whoa, it just kind of crushed him a little bit, right? And so essentially what happens is that reciprocal inhibition kicked in for him and it helped reduce the tension, which decreases performance. And so that's the other kind of key difference here between reciprocal inhibition and the stretch reflex. The stretch reflex kind of makes you more explosive, and reciprocal inhibition makes you less explosive. And so those are neurological kind of things that go on with stuff. Questions with those guys? Okay. So basic muscle physiology things. What you might want to do with this is you go back, and I posted the videos that you guys made of your little Play-Doh models. We all know that one was better than the other one, but you might look at both of them and kind of familiarize yourself with some of the basic structure and functions of those things because they were explained pretty well across both of the videos that you guys made. But your muscle fiber is just like a neuron in that it's a cell. A muscle fiber is basically the smallest, essentially, part of your muscle for the most part. And that's just a little strand that causes a muscle contraction to occur. Within your muscle fibers, then, if you have a bundle of muscle fibers, that's your fascicles. So a fascicle is a bundle of muscle fibers. And then a myofibril is the opposite. A myofibril is a tiny little section of your muscle fiber. And so it's a tiny little strand within the individual muscle fibers. So you might go back and look at the pictures that we had in our lecture notes that kind of showed the hierarchy of these different things. And then the sarcomere is one little tiny section within a myofibril. And the reason that we talked about a sarcomere is that the sarcomere is the contractile part of a muscle fiber. There's lots of stuff in a muscle fiber. It's got nuclei and mitochondria and all that kind of stuff you'd expect to find in a normal cell. The sarcomere kind of just limits it to the part that makes the muscle itself contract. And so within the sarcomere, then, you have a variety of proteins. We talked about 4 of them. We talked about actin, myosin, tropomyosin, and troponin. And I think you guys are pretty well clear on what those things do. Just remember that myosin is the big kind of the what they call the bigger, essentially, protein, essentially, of the sarcomere. And that one is the one that attaches to actin and then pulls it towards the center of the sarcomere. And remember that the myosin heads are kind of arranged tail to tail, and so they're kind of oriented away from one another. And so when they latch on and pull, they pull towards the middle, essentially, of the sarcomere. That's what makes that contraction occur. The double helix refers to the arrangement of the actin protein, and they're kind of wound around one another like little balls, basically. They're just kind of wound around one another. And then troponin and tropomyosin are part of the regulatory process of whether or not myosin will interact with actin or not. And so troponin is sensitive to, what's troponin sensitive to? What chemical? Calcium, right. So when calcium is released from the sarcomere, which I don't know why that's, it is, or no, I'm sorry, not the sarcomere. When calcium is released from the sarcoplasmic reticulum, which is down there below, then what does troponin do then when calcium is released?
It tells tropomyosin to get out of the way.
Right, it tells tropomyosin to get away from the binding sites or stop covering up the binding sites on those actin proteins. And so when that binding site is open, myosin then will latch on to the binding sites on actin and then begin the process of a muscle contraction. And so at rest, tropomyosin is covering up the binding sites on actin. It's preventing myosin from interacting with actin. And so the analogy we made would be, it'd be like 2 magnets. And if you have 2 strong magnets, but you put a piece of cardboard between them, they don't stick together. That's what tropomyosin. And tropomyosin is the cardboard between those 2 magnets. Once that cardboard is taken away, the magnets then lock onto one another, and that's what that's what myosin and actin will do, is they'll just naturally lock onto one another. And so that's the role that troponin and tropomyosin plays, that they help regulate if there's going to be any contraction occurring or not. And so the sarcoplasmic reticulum then, or the SR, is what that's the bag of calcium. That's its role, is to kind of regulate when calcium is released or not released. And so the signal that comes down from the muscle into the lower levels of where the muscle contraction is going to occur is carried by your transverse tubules. So the transverse tubules create a passageway from the outside of the muscle to the inside of the muscle to where the contraction is going to occur. And basically, for our purposes, the way that we explained it was that the transverse tubules will terminate at the sarcoplasmic reticulum, and then when a signal does come down there, then calcium is released. And so that all kind of goes back to our whole pattern of activity that occurs at the neuron. Signal goes across the synapse. It's picked up by a receptor on the muscle side. That signal then is carried down the transverse tubules to the sarcoplasmic reticulum and then causes calcium to be released. So that's the basic sequence that occurs within that stuff. And then the sliding filament theory is just the explanation or the name of the process by which myosin and actin work, is because the actin filaments will slide across one another when a contraction occurs. That's what the sliding refers to. That's probably the most confusing and complicated part of anything we've talked about. And what I would suggest that you try to do, if you can, is is kind of do it like I had you guys do with your Play-Doh models, is look at it from the neuron and the process that occurs from the neuron all the way down to the muscle and understand in basic terms kind of what are the names of the key players involved in that process, what are some of the chemicals that are involved in that process. And if you can kind of understand it from that top-down level, that's probably the best way to understand it. And believe me, this is a really pretty good simplification of what actually happens. There's a lot more kind of stuff that goes into place there, but that's enough to basically understand the main thing that goes on with a contraction and a muscle contraction. So, and I think the other kind of thing we tried to stress was the idea that it's a chain reaction of activities. Something has to happen that causes something else to happen, which causes something else to happen. And if at any point in time you take away one of those things, everything stops. And so it's a very regulated process that occurs, which is kind of interesting to consider. All right, should we talk about anaerobic fuel? All right, we've got two terms right here, ATPase and ATP. What did I tell you about any word that ends in ASE? It's an enzyme, right? And so ATPase's job is to split ATP into ADP. And ADP is essentially ATP that's missing a phosphate molecule. And when the phosphate molecule is ejected from ATP, it releases energy. And that's the energy that's used to fuel lots of different things within our body. And so ATP is sort of our universal energy donor. It's that main currency that's used to do lots of things in our bodies. Whether it's for running the pumps that pump sodium out of from within the myelin sheath, or if it's used to put calcium back into the sarcoplasmic reticulum or make a muscle contract, that's what ATP essentially does for us. So ADP then is adenosine diphosphate as opposed to adenosine triphosphate. That's just an ATP molecule that's missing a phosphate, and it's sort of useless to us, right? And so ADP is left behind after we eject that phosphate molecule. And so what we want to try to do on some level with all forms of metabolism is try to reform ATP out of these stranded ADP molecules that are kind of left behind after you release the energy from it. And so we'll get back to that in a second here. So fast twitch and slow twitch refers to what?
Muscle fibers.
Right, it refers to muscle fibers. And the main distinction between a fast twitch and a slow twitch muscle fiber is the type of ATPase enzyme that's located in the head of those myosin proteins. So fast twitch muscle fibers have an enzyme that splits ATP really, really quickly, whereas slow twitch muscle fibers have ATPase enzymes that split ATP at a slower rate. And so the neat organizational structure of our muscles is that all the muscle myosin heads within a given motor unit all have those same subtypes of ATPase enzymes. So they all become fast twitch or all become slow twitch muscle fibers within that particular motor unit. So the terms anaerobic and aerobic, we know those terms mean without oxygen or with oxygen, respectively. But what's the best way to consider whether activity is anaerobic or aerobic? What are the main kind of ingredients in those? Right. It's high intensity versus low intensity. That's the main way to kind of think about this stuff, is that if you think about no oxygen with oxygen, it's like it doesn't mean anything. But if you think about intensity or relate it to activities that have high or low intensity, that's what you're getting at. So anaerobic things are high intensity activities that can't be sustained for a long period of time. Aerobic activities are the opposite. Lower intensity activities that you can kind of keep doing for a long period of time. That's the main understanding of the distinction between those two things. So the next terms up here, the phosphagen system and the ATP-PC system, those are two names for the exact same system. They're not different things. There's two different names for the same thing. That's your first anaerobic system that comes into play. It's your short-term, high-intensity source of energy. How does it work? How does a phosphagen system work? And again, keep in mind that the role of all of these energy systems, the whole thing they're trying to do is they're just trying to reform ATP. And so how does the phosphagen system help reform ATP?
Takes it from creatine phosphate.
Right. That's right. So you have a creatine phosphate molecule, which is a creatine compound and a phosphate attached to it. And so the ATP-PC system, or the phosphagen system, essentially splits that phosphate molecule off the creatine molecule and then donates it to ADP, which then creates ATP. That's how that system works. It's a very, very short duration system. We said it lasts for not even 10 seconds of high intensity activity, but it's a great system because there's really no bad byproducts as a part of it. All you're doing is taking the phosphate molecules and adding it to ADP to make ATP, and there's kind of no downside to it. The downside is that it only lasts for a very, very short period of time. There's not a lot of creatine phosphate in our body at any given point in time. So creatine kinase, then, that's the enzyme that splits the phosphate molecule off of the creatine molecule. That's what its job is designed to do. So that's the first system to come online that's anaerobic. And then what about glycolysis? What's the funny thing about glycolysis?
It can be with either system.
Right. It can be either anaerobic or aerobic. Right. And so the distinction is that it's going to create one of two different types of acids at the very end, in addition to the ATP molecules that it creates. The high intensity or the anaerobic version of glycolysis creates lactic acid, whereas the lower intensity or aerobic version of glycolysis creates pyruvic acid. Remember that the way glycolysis works in general then is it takes sugar from your body, or glucose or glycogen from your body. Through a complex chemical process, then it's able to create ATP out of that by creating phosphate molecules that attach to ADP molecules. It creates ATP. But it doesn't create a lot for us. It only creates like 4 ATP molecules per unit of glucose that comes out of your body. So it's not a very efficient system. And in addition, in high-intensity activity, it creates lactic acid, which will eventually catch up with us and make us throw up in a bucket. Right?
Trash can.
Or a trash can. I'm sorry. Yeah, that's right. It was a big bucket. Right. Right. So that's the downside of glycolysis, is that it's a pay-to-play system. You've got to put in some ATP to make it work. You've got to put in some sugar to make it work. But the end result is that you do get some energy out of it. And so you can help sustain high-intensity activity with that system because the ATP-PC system runs out of gas after 10 seconds. So this system will give you another minute or so of activity before you've got to really kind of cut down on the intensity of the workout that you're doing. The significance then of pyruvic acid then is that pyruvic acid is the starter of the aerobic system. So the aerobic system is a two-step process as we discussed it. The first part is the Krebs cycle. We also call it the citric acid cycle, but the Krebs cycle and the citric acid cycle are the same thing. That's the first step in the two-step process of aerobic metabolism, or low-intensity production of energy. So pyruvic acid is dumped into that, and then we talked about, we mentioned it very briefly, but pyruvic acid is converted into a chemical called acetyl-CoA, and acetyl-CoA then is what kind of runs the Krebs cycle. The Krebs cycle creates electrons, and I've got them listed up here as NADH and FADH. We didn't get into that in a great deal in this semester, but those are the two electrons that come out of the Krebs cycle, more or less. And then those two guys, NADH and FADH, they go into step 2 of the aerobic process, which is the electron transport chain. The electron transport chain is able to leverage those hydrogen ions that are part of NADH and FADH, those electrons that are in there, and they're able to create, through a membrane process, they're able to create an imbalance in the positive side versus negative side, but that's kind of getting beyond the point. But essentially what the electron transport chain does then is it's able to create ATP very inexpensively for us. And so we get 32 units of ATP out of the electron transport chain per unit of glucose, whereas we only get 4 out of glycolysis proper. And so it's a really, really nice system that produces lots of energy for us at a very low cost for us. So the term oxidative phosphorylation that's on here, that's the collective name for this two-step process that includes the Krebs cycle and the electron transport chain. That's just the fancy word for that. Remember what the Cori cycle is? We discussed it only briefly. The Cori cycle's job is to suck up lactic acid and then convert it into glucose for us. And so it's part of the cleanup process. It's in our liver, basically. The Cori cycle is a process that occurs in our liver that vacuums up the lactic acid that we produce in glycolysis and then convert it into sugar that we can use later on.
Turns lactic acid into glucose?
Into glucose, into sugar, yeah. Which then we can convert back into ATP if we need it to. because we said that the lactic acid that your body creates, it can't just sit there. It has to be converted into something. So some of it's converted into sugar. Some of it is converted into pyruvic acid. So it's when your body eventually rests after you've thrown up in the bucket, in the trash can, your body can then start to do something with that lactic acid. And that's part of the reason why you want people to move around when they're done working out, is you want their blood flow to keep moving so that your body it helps your body clean up all the waste products that were created when you worked out really, really hard. Because when you just lay on your back when you're done with the workout, that's not a good way to do things, because your body can't pump blood around very easily when you're laying down. Okay. So up to that point, that's probably the hardest stuff that we've gone through. And so what you'll want to do is look through your notes, and then, like I said, I made relatively short podcast recaps of those lectures. I tried to make them very much to the point, and so you might listen to those too, just to kind of refresh your memory on some of that stuff. If you can get that stuff down, you'll probably be in good shape on the exam, because the rest of the stuff, you'll know enough and remember enough of it that you'll probably be able to work your way through it. All right. And so I would focus your studying on that stuff. The rest of the stuff should be relatively straightforward, but let's run through this stuff quickly anyway. So carbohydrates, proteins, and fats. How many calories do we get per gram from these things? How many do we get out of carbohydrates? 4 calories per gram. How about proteins? 4 calories per gram. Fats? 9 calories per gram. That's correct, right. So that's what you get out of those things. So fats are a very, very high energy source, right? That's one of the reasons why people have high-fat diets. The problem with that is that you're also having a high-calorie diet along with that. That's the problem you run into with fats. But which of these 3, then, is your preferred energy source? Carbohydrates are, because carbohydrates are most easily broken down into glucose or glycogen and are thrown into glycolysis to create energy for us. So the quickest source of energy for us are carbohydrates, essentially. And then what's second place behind carbs? What's our second preferred energy source behind carbs? Fats, that's right. So fats come in second, and then proteins are in third, essentially, behind us. The 3 different types of carbohydrates are monosaccharides, disaccharides, and polysaccharides. It's just from low complexity to higher complexity types of carbohydrates. The terms glucose and glycogen are just names for sugars. And so glucose is a monosaccharide. It's a very, very simple sugar that's easily broken down. Glycogen is a polysaccharide. It's basically hundreds or even thousands of glucose molecules all slammed together in order to kind of consolidate space within our bodies. And so when we eat anything, we can either convert that food down to glucose and use it immediately, or we can convert it to glycogen and then store it in our bodies for later. Sucrose and fructose are just different types of sugars, and so sucrose and fructose are both disaccharides, or just combinations of sugars. And then we have different types of fats. We've got fatty acids, triglycerides, phospholipids, and steroids. Which 2 of those are energy sources for us?
Fatty acids and triglycerides.
That's right. Fatty acids and triglycerides, those are the energy sources for us. And all triglycerides are groups of fatty acids tied together. So you can kind of think of triglycerides being a little bit like a disaccharide, and that's a disaccharide is groups of sugar molecules attached together. That's sort of what a triglyceride is. It's fatty acids then. Instead of sugars, it's fats that are tied together, essentially. And then what's the main function of phospholipids? It's not an energy source. What do we use phospholipids for? Phospholipids are used for cellular membranes. And so the membranes that are around cells and around like the myelin sheath, that stuff is made up of phospholipids. That's where we get that stuff from. And then steroids are another type of fat. And what's the main function of steroids? Right, hormone production. That's exactly right. Yep. Then we got into some oxygen kind of related stuff. We talked about METs. I don't think there's much on this exam about METs, but a MET is basically by definition, a MET is 3.5 milliliters per kilogram per minute of oxygen you're using. I won't have this on this exam because we didn't do our VO2 max lab yet, and that's when that stuff will kind of make more sense to you, and so I'll make sure if there's anything on that, I'll take that off. But do you know what VO2 max is? That's just a term that refers to what is the most oxygen that you can use when you do any kind of physical activity. And the reason that we want to look at that term is that that's what the pacer measures. The pacer, when you enter your data in, is measured in VO2 max. It says how much oxygen can your body take up and use based upon your body weight. And then we drew those graphs that looked at oxygen deficit, steady state oxygen debt. You should know what those three terms are and probably be able to identify them or draw one of those graphs like we had. Lactate threshold is one that confuses students sometimes. Lactate threshold is basically the intensity that you work out at, that when you get to that point, you start producing lots of lactic acid. And then that means if you get to that threshold, it means you're not going to be able to maintain that activity or that intensity for much longer. And so a lot of endurance athletes want to know what their lactate threshold is, and they want to kind of, I just saw that there's a marathon that was run recently, I want to say, and the guy that won it, over the course of 26 miles, his average mile pace was 4:27, which is ridiculous, right? But for him, yes, every mile was at 4:27. That's right. And for him, he is so efficient and such a good runner that he's able to run at that. I mean, for us, that would be almost a sprint, right? That'd be like you'd be full out, right? But for him, he's in such good condition that that's still an intensity level that's low enough that he's able to stay below his lactate threshold, right? Whereas for us, we couldn't even physically do 1 mile that fast, right? And even running one lap around the track at that pace would be very painful for us, essentially, right? But it's all relative, and that's kind of the concept here, is that relative intensity refers to how hard things are for you when you're doing them. Ergogenic aids, this stuff was pretty straightforward. Placebo effect just is essentially looking at, you may not be getting the actual drug, but you think you are, and so all of a sudden you gain the benefits of it because your mind is so strong it overcomes the fact that you're not actually getting the real thing. A blind study is one in which the subjects don't know if they're getting the real thing or not, so you're trying to hopefully eliminate the placebo effect because you can kind of compare the control group versus the group that actually got the real stuff and see who actually did better. And then a double-blind study is one in which the subjects don't know if they get the real thing or not, and then the researcher doesn't know either. And so there's no research bias to go along with that. What do blood doping and EPO have in common? What are both of these things designed to do?
Increase oxygen in the blood. Red blood cells.
Right, increase your red blood cells, which then will increase the oxygen in your blood, right? So blood doping is where you take blood away from somebody, you separate the red blood cells out, you re-inject the rest of the stuff back into their body, and then you store their red blood cells in a freezer, and then when they're getting ready to compete, you re-inject their red blood cells, and then they're superhuman and they can win everything. What's the problem with blood doping? You could die, right? That's the real risk. Right, because your blood becomes so much thicker because it's full of extra cells. And then EPO then is just a drug you take. So instead of having to draw blood, separate red blood cells out, and then re-inject them, EPO just forces your body to create more red blood cells on its own. So those two techniques would be good for aerobic athletes. So any kind of endurance athletes would benefit from that kind of a thing. Blood buffering is then the opposite. So blood buffering is when you take a bunch of stuff that helps counteract the lactic acid your body creates when you work out really hard. We said the problem with that is that the amount of stuff you have to take in order to actually do that might make you really sick, and you won't be able to perform very well. And so it's, but the reason I brought blood buffering up is that it kind of is designed to kind of work for the opposite kind of athlete than blood doping. Blood doping is more for aerobic athletes. Blood buffering would be more for anaerobic sorts of athletes, so like wrestlers or sprinters and stuff. pH is just how we measure the acidity of your blood. And then we just got a couple last ones here. Testosterone is the main male sex hormone, helps increase muscle mass and increase aggressiveness and things like that, right? Estrogen is the female sex hormone, does the opposite thing, makes people really nice and friendly, except for my wife who's mean. And then the term stacking and cycling. Stacking means you take a variety of drugs on top of one another, and then each of those drugs then hopefully increases the effectiveness of the other drugs that are around them. And so it magnifies the effect of each other. Cycling then refers to going on a schedule where you take some drugs, then you go off them for a while, then you take them again. So the idea behind cycling then is to reduce your body's dependency on whatever drugs you're taking. Because the big concern for anyone that abuses anabolic steroids is that they mess up their whole body's hormone production process. Is that all of a sudden a guy that might be taking it, if he abuses anabolic steroids and he goes off them, his body just has stopped making testosterone. And that leads to some weird issues as far as how his body will function, because now estrogen is at a more dominant level compared to testosterone. And then the opposite thing can happen for women as well. Growth hormone, the last thing that we kind of talked about was what's the main difference between what does growth hormone do differently from testosterone? Right, it makes everything build up and get bigger. So testosterone primarily will just affect your muscles, whereas growth hormone will affect your bones, will affect your muscles, will affect tissues, it'll affect the size of your internal organs. Growth hormone is really scary, and so it's kind of nice that some sports are starting to test for it, because it's kind of done some weird things to people. And not that testosterone's any angel either, but any kind of drug abuse can be bad, but growth hormone is kind of creepy in what it does. Growth hormone is just harder to test for. That's why a lot of athletes were using it, because you can't pick it up in a urine sample as easily as you can a blood sample.